Well, that's only, that's a, well. I don't think it's so.
It isn't. Well, hello everybody. Welcome to our program meeting for this evening. I might mention one thing, and that is to remind everybody to put their cell phones on vibrate or silence them somewhere. Let's see, a couple of announcements. I guess the first one I'll kick off with is Tom. I think Tom is heading up our nominating committee, and so he's going to tell us our nominations and then also issue a plea.
Yeah, so we thought we had this all settled, but it turns out we have one position still we need to fill for the next year, board members. Kevin has agreed to be our treasurer, which means our vice president position is open, and we are still looking to fill that position. Barbara Gubbard has agreed to sign up again as secretary. Mark Nolan as on the board of directors, and we've asked Jennifer Brewy to be a nominee for the board of directors. So I can't say about the vice president right now. We're still looking, but we can make nominations from the crowd next.
If anybody wants to.
We're looking for volunteers.
Thank you, Tom. We'll be voting on the slate of officers and directors at the next meeting or maybe. And let's see, I wanted to also mention we've got a couple birdathons going on, or maybe three. Tom, are you doing a bird? You're doing a birdathon,
Yeah.
aren't you?
Yeah.
Okay, so Tom is doing a birdathon. I think that's local, Sedgwick County. Do you know what date you're doing that yet? Not yet, okay. We've got a couple others going on. Another is the Chaplain Nature Center team is going out. And we're doing ours on the 6th and 7th. We kind of do 2 half days, Friday afternoon and Saturday morning is kind of how we do ours. So, and then Patty has got one going on. I'll let her mention about that one.
Carol and I and whoever other women we can grab together are going to go out. We're the Painted Funding, so we're going to go out on Friday, May the 6th. And we're the only ones that are efficient enough to have pledge sheets for tonight. So here, Jim, pass these around. I wanted to mention one other thing. I just noticed yesterday that the Walk with Wildlife, which is the big event for the Great Lakes Nature Center, is on May 14th, which is the same day as our second Saturday program. So I talked to Amanda, and it will be okay if we meet in the west parking lot off of Oliver and do our walk over there. Their Walk with Wildlife will just be up here on the Quail Trail, which is this closest to the building. So I put it in the newsletter, and we'll have to remember to put it on listserv and Facebook and everywhere else to remind people. But next bird walk will be on the west side.
Thank you, Patty. I guess one other field trip to announce on May the 7th, that's Saturday, Woodson County Spring Migrants. And so Mark Nolan is leading that one, meeting at the Dillons on on Kellogg, and that's the end of the place we met for the last field trip. At 6:30 in the morning, he's going to wander around Woodson County looking for migrants. And then also I'll mention our next program, the May program meeting. Laura Mendenhall will be here talking about the backyard habitat program. So that'll be a good one for if you're interested in what to plant in your backyard to attract more birds, that sort of thing. So but anyway, All right, at this time, I'm going to go ahead and introduce our speaker for this evening. Emily Ostrow is our speaker. She's going to tell us some really interesting things about great horned owls, probably some things you didn't know. Emily is from Philadelphia and got her bachelor's degree at Drexel University and is currently earning a PhD at KU. So I'll turn it over to Emily.
Thank you so much. Thank you so much for having me here today. I'm really excited to share some of the work that I've been doing. So, as Kevin said, I am doing my PhD at KU in the Ecology and Evolutionary Biology department. My research focus is generally on birds and bird genetics, so hopefully I will be able to effectively explain some of the genetic analyses that I've been doing. So yeah, I am talking about great horned owl diversities at 3 different scales. Throughout the Americas, within the US and Canada, sort of North America, and also within Kansas. Before I talk about great horned owls, I want to talk about sort of natural history museums. I am associated with the Natural History Museum at KU. And so natural history museums, when you think of them, there's often a public space where they have exhibits, but there's also usually a collection where we have study skins. And so study skins are incredibly important. It's maybe a little hard to see here, but the bird that is in this photo is collected by Alfred Russel Wallace. You can see that right here. And so specimens have important historical significance and also are incredibly useful to tell us about birds in ways that you can't get by looking at them in the wild. So that was a historical specimen, but modern specimens record many types of information. So you can have information about internal anatomy, what their plumage looks like, and also other associations such as parasites. So the specimen that I have here is of a great horned owl, and most of the owls that I'll be talking about in this talk have been donated from wildlife rehabs who couldn't save the birds or have been found on the side of the road as roadkill. 4 of the owls that I used in my project are actually from here, which is cool. So that's where I'm getting a lot of the birds that I'll be talking about today. And so what you can get from these birds is many types of things. We have ecological connections with gut contents. You can keep physical attributes for years. I'll be talking about some genetics, and you can also get some information on parasites. So there's one of the parasites I'll be talking about with a tiny bead brush that you pick the parasites up with. And basically we put them in little tubes of ethanol, and that's how we keep the parasites for some of these birds. So the first scale that I will be talking about is great horned owls, sort of range wide. Now, something that I was not super familiar with growing up in the Philadelphia area is that there's a wide range of phenotypic diversity in great horned owls. So I sort of grew up in the middle of this eastern group here. Okay. In the middle of this eastern group, so I didn't really understand the breadth of plumage characteristics in great horned owls. But this photo on the left here shows some of the variation. You can go from a very dark individual in sort of the Pacific Northwest to a bird that looks almost like a snowy owl in sort of Canada area. And so before my work, there has been there's been 2 studies on sort of the genetics of great horned owls. This is the more comprehensive one. This is based on 1 gene. And so if you're not familiar with this type of graph, can I pick this? Yeah. Sorry, I like to move. So basically, this graph is showing a few individuals. This is one individual, this is another individual, this is a couple. And basically this is showing two changes between two bases, like in DNA, between this individual and this larger group here. So these are some subspecies that are found in Western North America. And basically what you need to know about this is that there doesn't seem to be any sort of larger pattern within subspecies. So an individual from this subspecies is right here and all the way over there. So there isn't sort of any, what we would call genetic structure within great horned owls at this scale. So the first initial question is, how genetically different are great horned owls? And there is an expectation based on some previous work that the Magellanic horned owl in South America might be a different species. So based on some nuclear data, sort of the typical type of data that you would think about when you're thinking about genetics, there are two sort of different groups, as you would sort of expect. So the way to read this graph here is an individual that is close to another individual is more closely related. So these two birds are most closely related to one another, but this bird is pretty distantly related to any of the birds at the top. And so there's two major groups here. There's this group, and then there's that group. So this is sort of supporting the idea that there's two different species within great horned owls as we know them right now. The mitochondrial data show a similar story, but is slightly interesting. So the mitochondrial data, if you try to think all the way back to high school or college biology, mitochondrial data is typically inherited through your mother. So it's a little bit different and it tends to evolve a little bit faster. So what's interesting here is that there is one of these southern South American owls found in the northern group. And that just shows that there's some sort of connectivity still between these two species. Now, what do these genetics say in context of sort of the morphological variation and local variation? Within the context of the sort of morphological variation, the Magellanic horned owl is not incredibly different outside of the range of the variation in North America. But what's interesting is the Magellanic horned owl here in gray is incredibly vocally different from anything else. The northern great horned owls have sort of 3 or 4 notes typically, and the Magellanic horned owl has 2 notes and then sort of a trill at the end. So sort of some conclusions for thinking about the range-wide diversity of great horned owls. This Magellanic species should probably be considered a full species. There's evidence of some sort of sharing historical genes or hybridization though, and there's very little genetic differences within the northern group at this scale. So then the question is, how are the genetics and subspecies related across North America? So just to pull up this subspecies map again so that you can kind of picture it in your head, there's quite a bit of variation in subspecies throughout North America. But all of this subspecies description has been based on morphology. So we know almost nothing about the genetics. And if you put this map next to a map of humidity here, a lot of the subspecies descriptions sort of match with different humidity variations. So there's sort of an eastern group, and then you get more diversity throughout the west, as there's different levels of humidity. So the genetics of great horned owls are actually quite different from the subspecies as we recognize them. So the genetics here, just think of sort of different populations being these three different colors. So there's an eastern group, there's a southwestern group, and there's a northwestern group. But one thing I want to point out that the other map doesn't really show is that these populations are very connected. So this graph is a PCA. It's kind of similar to the phylogenetic tree that I showed earlier, where you can interpret this by looking at the closeness of the dots. So these two individuals are very closely related versus this individual is not very closely related to this individual. So you can sort of see three populations like the last map showed, but there is quite a bit of integration between these groups. So the question is, are these groups true populations? This map here is sort of trying to address that. And so this map is it's describing a principle in genetics called isolation by distance. And what isolation by distance is that if you're an owl in Florida, you probably won't mate with an owl from Alaska. It's just not physically possible. But that doesn't mean that you're different because you can't breed together, or you're different because you're more evolved to be in that specific environment. That just means that you're separate, so therefore you can't mate. So we expect that there is a certain amount of differences just due to the fact that you are far away from another bird. So this map here is showing areas where you are more or less likely to have sort of a barrier in mating versus just the expectation that if you're further away, you might be slightly different. So any of the areas in darker blue, you are there is less genetic, or there's more genetic migration than you would expect. And any of the areas here in dark orange, there's less genetic migration than you would typically expect. So putting the map from the first map that I showed you onto this sort of map of genetic connectedness, you can see that there's sort of the genetic barrier between the eastern population and the western populations, which makes a lot of sense if you think about sort of a lot of the bird distributions. There is a pretty distinct transition in many bird species between eastern birds and western birds at both the species and subspecies levels. So within North America, great horned owls are phenotypically diverse, but exhibit only 2 to 3 non-isolated populations. Phenotypic diversity may be based on environmental factors such as humidity, but they're not necessarily based on different breeding populations. And the geographically aware analyses of isolation by distance identify a slight genetic barrier between the eastern and western populations. So my next part of this talk is zooming in looking at this eastern and western transition, specifically in Kansas. And I am bringing in a little bit about parasites, which is a little bit of a personal interest. So I hope it's interesting for you all as well. So previous to this, I have been talking about the diversity throughout the range of great horned owls. But there's actually a lot of phenotypic diversity within Kansas itself. So this picture in the upper right-hand corner shows just the variation of great horned owls that you can see in Kansas. This is a transition between two subspecies. So Bubo virginianus pulescens is in western Kansas, and like it's called, it's a pale kind of sandy colored bird. And then Bubo virginianus is in the east, and it is a darker and sort of warmer, redder colored bird. Now, one thing you might not have thought about before you came to this talk was that a lot of birds have a lot of parasites. And so throughout preparing many of these specimens, I found that approximately 90% of the owls that you would see out and about have parasitic lice on them. So parasitic feather lice are highly evolved with birds themselves. So lice have to be on a bird in order to survive for most of their life. So basically the way that they move from host to host is one bird touches another and then they move. They are not often found off of birds, and they cannot survive long outside of the sort of humidity and environment of a bird. Another thing that's interesting is that these lice tend to evolve to match the coloration of the birds that they're found on. So this skull here has sort of a white gloss. It might be hard to see in the back, but the body of the louse extends to here. So sort of the second half of its body is white. And this juvenile American robin has many tan lice on it. So at least at the species level in the wild, there's a lot of sort of color matching between parasites and the birds that they're found on. This study of Galapagos hawks kind of shows that this sort of close connection between the parasites and the hawks that they're found on, or birds in general, shows that there's a lot of genetic matching between the hosts and the parasites. So this graph is a little complicated, but what I want you to take away from this is each of these bars here is an individual. And this is basically showing that each of these colors is sort of a genetic population. And so the populations of the lice are extremely similar to the populations of the birds that they're found on. Yes.
Going back to you had those series of owls, and the east and west is different. You call them subspecies, or are they phenotypic type? What's the difference genetically between those? Is it just skin color or feather color, or?
So I'm going to get into that later,
Okay,
but
okay, fine.
throughout, sorry.
No, that's okay, that's fine.
Throughout North America, there isn't much genetic differentiation. All right, so we know that lice and owls are very closely mirroring each other for what their populations look like. This is an experimental study done on the coloration of lice and how quickly they evolve to match their hosts. And basically this study has shown that within 4 years, the coloration of their lice can evolve to match the coloration of their hosts. So they this study put intermediate colored lice on both white birds and dark birds. And just over 4 years, the coloration evolved to match either the dark or the light birds. So there's sort of an interesting pattern of both genetic associations and color associations in different studies. So my question was, how much selection for color is there in natural populations? So as I described before, the eastern birds are in general darker. The western birds are in general lighter. But one thing I want to point out is that there's a lot of individual variation. So some of the birds in the west are going to be fairly dark. Some of the birds in the east are going to be fairly light. So I have a few sort of expectations based on what we know about this association. So we talked about the messy hybrid zone here. And so the other point that I want to make is that lice are typically transmitted vertically. Vertical transmission is basically just the idea that lice are transmitted from parent to offspring. So, like I said earlier, lice are typically transmitted through physical contact. So they can be transmitted from mate pairs, but a lot of this transition goes from adults to the chicks that they're taking care of. So basically, any individual bird can be exposed to lice of any color. And so there's different expectations that you can have based on different levels of selection for color. Under no selection, you would expect that light lice can be on either dark or light birds. With some selection, you would expect that last populations that arrive on a host eventually adapt to host coloration. So eventually these lice are changing to their host coloration, but there isn't any sort of immediate removal of these individuals. Under strong selection, lice populations that don't match their hosts are just immediately removed. And so they don't sort of contribute to the genetic population on the birds themselves. So under different amounts of selection for color, you would have different expectations for whether the last coloration or the last genetics match their host. Under no selection for color, you would expect that last coloration would not match, but that their genetics would. Under intermediate selection, you would expect that both color and genetics should match. And under strong selection, you would expect that last coloration matches their host, but last genetics might not. So to address these sort of situations, I collected both genetic and color data from lice and owls. Unfortunately, I only have the colored data and the genetic data from the owls to share today. So this is a map of sort of the sampling of owls that I got from different sources, and it is colored by what species of lice were found on each of these individuals. So there are two species, Strigiophilus oculatus and Strigiophilus cyrni. And so Strigiophilus cyrni is found throughout this transect, but interestingly, the Strigiophilus oculatus species is only found on either end. So I needed to add a few pictures of owls in here. So this is sort of a description of how I got some of this color data. So this is called quantitative imaging. And basically what it is it is a fancy way to sort of simulate the outside environment. So these are special light bulbs, also known as reptile lights, that imitate sort of the sunlight. And so they have a certain amount of UV in them as well, which is not used in typical lighting. And you take the photos of these birds with this color chart here to sort of standardize all of the photos. I did the same thing with lice. So I took photos of lice with this microscope. This microscope takes 200 photos of each individual louse and basically takes different amounts of focus for each of these photos so that you can get this nice sort of very clear photo on the right here. And so with those photos, the louse and the bird photos, you can have a nice photo of both the front and the back of all of these individuals and information about the color as well. And so with each of these photos, I chose regions where I took the color from. So it's estimating colors from each of these areas here and kind of creating an average. So before we get into some of the color data, I want to share sort of what we know about the genetics. This is the same type of plot as I described earlier. This is a PCA. So just as a refresher, these two individuals are very similar to one another versus this individual is pretty different from this individual. So what you can see here is, and the dots are colored by longitude. So Western individuals are blue, and Eastern individuals are red. So what you can see here is a pretty clear transition from genetically from Eastern individuals to Western individuals. Now the coloration of the owls, like I kind of showed in the pictures before, is not as clear. There's still sort of a general pattern from west to east, but you have some Eastern individuals that look like Western birds and some Western individuals that look like Eastern birds. Now, maybe a more intuitive way to think about this is looking at it sort of geographically. So the axes here, the y-axis is the proportion that you would be a Western bird. So if you're at 1, you're a very Western bird. If you're at 0, you're a very Eastern bird. So looking at the genetics and the coloration, you can see that there is a pretty consistent transition in both groups. But one thing that might be a little bit more subtle is that there's a sort of sharper transition in the genetics than in the coloration. So say you were here in Wichita and you were to look at birds in your backyard. The birds that you would see would be sort of intermediate between Western and Eastern individuals. But the genetics are actually much more Eastern than Western here. So this graph is the two graphs that I showed you previously, sort of combined on top of each other, with the addition of individual single nucleotide polymorphisms. You can think of these SNPs as sort of like genes. So they are basically independent genes that are sort of transitioning across the state. And so this genetic background is telling some of the story, but individual genes can be transitioning much sharper or less sharp than the genetic background. Now, comparing what we know about owls to the coloration of the lice, there's actually not quite as much of a pattern in the lice based on whether you're an Eastern louse or a Western louse for either of the species. This is sort of another way to look at this association. So basically the way that you read this graph, this is a correlation plot, is that if any of these dots are larger, that means that there's a high association between any of these variables. If any of these dots are smaller or lighter, that means that there isn't much of an association. So this is a little misleading because all of these comparisons are within just the louse individuals themselves, and all of these are comparisons are between the birds themselves. So if you just look at the comparison from louse to birds, there doesn't seem to be a lot of association between the coloration of owls and lice. So basically what you can take away from this part of the talk is that parasites generally have a close relationship with their hosts and are often affected by the characteristics and behavior of their hosts. Great horned owl coloration exhibits sort of a messy transition between these two subspecies across Kansas, but the genetic populations have a comparatively sharp transition. Parasite coloration does not seem to be affected by host coloration, but theoretically, hopefully in the future, if you see me talk again, I can share a little bit more about the genetics of the lice and how they're affected by the birds. Thank you very much. Does anyone have any questions at this time? Yes.
So how different genetically are those different variations of the owl? Is that kind of a subspecies, or is that just a type variation, a physiological type or whatever?
Yeah, so the variation here is, I wouldn't even call them subspecies. They're incredibly closely related. I think it's just a lot affected by the humidity and the environment that they're in. So that could be the difference of a few genes, not necessarily sort of different populations.
So they can interbreed
Very freely.
very freely.
Yeah.
And it doesn't make any difference on coloration between two different owls as far as interbreeding, so. And that's the definition of, is that still the definition of species and subspecies, or have we gone beyond that since back in my day?
Well, there's a lot of disagreement about what a subspecies is, but typically there's some sort of preference for like your own subspecies in breeding. Like obviously there's interbreeding between subspecies, but I was reading a while ago that sort of 80% of your genes should be shared within your own subspecies to be considered a subspecies. That's a very loose definition, but basically there's some preference for your own subspecies. And this does not seem to be that case.
So that's for owls, or for birds, or for?
For owls, yeah.
So what's the generation length for the lice?
So
A year, or how long what's the generation for them?
that's a good question. I used to know that answer. It's maybe a month or two. So they're evolving much more rapidly than the birds.
So 4 years is probably
Many
50
generations.
generations.
Yeah. Yes.
Yeah, how long did these studies take so
For me to do,
far? Yeah.
this has probably been about 3 years of work. Yeah, I didn't know what I was doing my first year. Yeah.
Your map where you had running the humidity.
Yes.
Kind of mimicked your other map. What made you choose humidity rather than altitude or temperature range? Any environmental statistics?
Yeah, so obviously all of those things affect animals physiologically, but there's sort of expectations that people wrote down like 100 years ago that it's called Glover's rule, where if any animal is in a more humid area, they're more likely to be dark. And if any animal is in a drier area, they're more likely to be light. So many of the, it's not. I tried to bring that up. But many of the subspecies that are in these drier areas are lighter, like you would expect. Yes.
And you would assume that since the owls don't build their own nests, and they use nests of previous birds, that transmission of lice could occur in the nest to the young ones in particular, and then to the adults, or vice versa. The two species of lice that you showed, are they just restricted to great horned owls, or do they occur on other species whose nests they might use them, such as red-tailed hawks or crows?
Yeah, so that's a great question. So the one species that's found on sort of either end, but not in the middle of this transition is great horned owl specific, but the one that's sort of found across this area is found on great horned owls and strix species, so like barred owls and things like that. So there are many barred owls in the area, and so there could be sort of sharing between great horned owls and barred owls, which might sort of confuse the coloration pattern in the lice. Yes.
So you said that when lice get on a host, that they quickly evolve to match the color of the host. I was wondering if you could just hypothesize how that would happen. Like, are the birds seeing the different colored lice and picking off the ones that don't have it?
Yeah, that's exactly what it is. For most lice, the birds can see the lice, and so they're more likely to pick a black louse off of a white bird than a white louse off of a white bird. There's specific lice that are found on the head that don't have that sort of color selection. Yes.
Aren't there a few subarctic specimens from Kansas? Seems to me I saw that somewhere recently, but I can't remember the context.
Yeah, so Subarcticus does definitely get down here. I don't think we have those specimens specifically here, but that subspecies does get down here. Yes.
I kind of had that question too. I was under the impression that the great horned owl would migrate from up north and down here, but that's a different subspecies. And you're saying these live here all year then, basically? Live in Kansas all year?
Can we get this working? I have a map.
I don't know, it's still on your window.
Sorry, I have a map that like addresses this question precisely. Well, I guess I can show you afterward, but there is a decent amount of movement in all owls, sort of, eruptive behavior, but most of the movement in great horned owls is sort of in this subarctic species that sort of migrates down and migrates back. And that's from bird banding data. So basically these are recaptured birds. Yes, here we go. So if you're sort of in a more southern location, you have less reason to move in the winter. But there is quite a bit of movement from some of these northern populations. And so when I did the study, I did specifically try to get birds that were more closely in breeding timing than sort of any random owl that's around.
So the motion, the northern ones up in Canada, they will move south during the winter, down to the United States, down to Kansas and
Yes.
Nebraska and that, and then they move back up in summer.
Yes.
Is that kind of food and rearing young, or?
Yeah, so they'll move out during the harshest part of the winter, but then they'll move back for breeding season. Yes.
How many species of owl lice did you say there are?
So on owls specifically, there are 4 species of lice. 2 of them were more common, which is why I focused on them.
Do other prey items, prey birds, are any of those lice capable of living on the owl?
So for short periods of time, maybe, but one thing that I didn't sort of touch on here is that size plays a big role in whether a louse can stay on a bird as well. So many of the prey items, the birds are smaller, the lice are smaller, so the lice wouldn't necessarily be able to survive on an owl.
So they're very host specific, like with mammals, oh, like fleas that inhabit dogs, they're not gonna stay on humans because the spacing of the hairs and the amount of hair and all that is real specific. So they can't live on cats either, so that's the same way with birds, with owls.
Yeah, it's similar. There are specific species of lice that are able to go on more species, or there are pretty specific species, but in general, like the difference between an owl and a songbird that they might want to eat is pretty different. So there's not sort of a lot of sharing of lice between owl and prey.
I'm tempted to think that if you're a host species, that all lice are bad. Is there any situation in which the lice benefit the host?
There are no situations I'm aware of where the lice benefit the host. Most of the time, it's pretty neutral. If there are many lice on a bird, then that can be very bad for the bird. There can be damage to the feathers. But most of the time, it doesn't seem like the lice are really affecting the birds that much.
And do the lice spend almost all their life on the bird, or they like ticks that they'll feed and then drop to the ground and then come back on, or do they spend all their life on the bird?
Yeah, so lice generally will stay their entire life on a bird. Sometimes if they drop off in a nest and another bird is like there within a few hours, then they can switch hosts that way. But they're typically living their entire life cycle on a bird. Yes.
How strong is this association of lice going from parent to child, to small bird? And what happened, and are the colors, I mean, I guess if they mate with a different color, then the lice, that could be bad for the lice.
Yeah. Yeah, exactly. So the association is pretty strong where basically if the birds need to be touching one another for the lice to be transferred most of the time. So there's a pretty strong association between at least familial groups where the lice are sort of transferred between birds that are related to one another or birds that are breeding with one another. And yeah, so if there is strong selection for color in lice, then that means that the lice that don't match the birds are being removed pretty quickly compared to the ones that do. It doesn't seem like that's necessarily the case in this situation, but in other species, maybe that's different.
All right, well, thank you very much. Thank you all for coming, and hope to see you next month at our meeting with Laura Mendenhall. So, thank you again.